I'm Tommaso Sbrana. I'm the CEO of Ivytech, and I'm a biomedical engineer. In the last 12 years, I focused my attention on the development of new platform to overcome the limitation of the standard multiple plate system or the transfer system. At the beginning, I focused my attention on the design of these new in vitro platforms when I was a PhD and a researcher at the University of Pisa. And then in 2014, in a team composed by 3 of my colleagues and myself, we decided to found Ivytech to bring to the market the solutions that we developed at the university. So I switched my attention to the business aspect of this new platform. So in this presentation, I would like to give you our receipt in order to develop an advanced in vitro model. So the investment in the preclinical phase of a new drug development are very high in money and in time. But considering the low success rate, we have a waste of money. And moreover, we have also to consider that company is legally liable for all the products released on the market. Therefore, our preclinical phase should be well correlated and predictive of the human reality. But here we have 2 main problems. The first one is related with the standard in vitro models. These models use a very old technology, which shows some limitations if it is used to mimic a very complex scenario such as the human reality. In particular, the multiple plate and the transfer are not able to mimic the dynamicity of the human reality, and also the crosstalk between the tissues, so the cross modulations of the tissues is not so easy to be simulated. On the other hand, we have the animal models. The animal models are ethically controversial, but we are not talking about these aspects in this presentation, but they are expensive if compared to the in vitro models. The range of expense is between 2 and 30 times more than an in vitro test. So a good strategy to reduce the cost could be the increase of the predictivity of the in vitro model in order to reduce the number of animal tests. And in order to reach this goal, we need an upgrade of the technology and the methodology that we use in order to build these new advanced in vitro models. So here we have the reality that we want to mimic. The human reality is a 5D scenario because we are in a 3D topology. If we consider a tissue, a single organ, an apparatus, or the whole body, it's a 3D structure. And this structure is in a dynamic environment. The dynamic environment means that we have a response to a stimulus, which is a time-dependent response. Moreover, we have at least the blood flow, which brings nutrient and fresh oxygen over the time and stimulate the cells because of a mechanical action. You have to consider that a liquid which is flowing in a certain volume is going to act with a mechanical action onto the inner layer of this volume. If you consider to have the cells onto this layer, you will have a mechanical action onto the cells. This mechanical action is described by a parameter which is called shear stress, and the shear stress is the stimulation of the cells to work in a certain manner. Moreover, we have a cross modulation between different tissues, so the response to an exogenous stimulus is the result of a cross modulation of different organs. The standard in vitro technology allows to study a 3D structure because we have on the market several scaffold or hydrogel solution in order to build our in vitro model in 3D topology, but they are not able to mimic the dynamicity of the environment because they use a static plate. Moreover, it's not so easy to have organ organs crosstalk study, because if we are not considering the co-culture, we are not able to simulate an exchange of information between different tissues by an exchange of liquid. The example could be liver, which communicates with the heart. So we are not able to mimic this kind of a scenario with a standard plate. So in order to build our advanced in vitro models, we have to follow certain rules. Let's focus the attention onto the 3D structure. We know that a single cell or cells in general are different in topology if compared to the standard 2D culture. They have also a different metabolism, and also the response to an exogenous stimulus is completely different if compared to the 2D scenario. So we have to understand how to build our 3D structure, and here are some guidelines. So we have to find a functional unit, and in this in the papers I reported in these slides, the researcher decided to use the rule of 10. So a functional unit, so the minimum amount of the 3D structure that we have to use in our in vitro model, will be composed by a cube, an ideal cube made by 10 times the cell diameter. So each layer will be 200 microns at least. Moreover, we have to decide how many functional units we need in order to be well correlated with the human reality. So we decide to use at least 10 functional units, so 10 cubes of 200 microns per layer. Finally, we have to decide the minimum amount of cell density. In this case, a good strategy is to choose 1/10 of the physiological density. So these are the minimum parameter. These represent the minimum parameter in order to build our 3D in vitro models. Once we have the mathematical approach, we have to choose the suitable structure in order to build the 3D model. So on the market, we have different solutions. So we have scaffold represented by porous sponge, gel scaffold, so hydrogel, and reconstructed tissues. In the last case, the reconstructed tissues are mature tissue that are sold as a ready-to-use product to test a cosmetic compound or, in general, a drug. In the first two cases, we have to understand which parameters we have to follow in order to make our choice. So the key decision points are represented by the stiffness of the scaffold. So we have to mimic the stiffness of the real tissue. The stiffness is dependent if the tissue is in physiological condition or in pathological condition. Moreover, the topology of the scaffold could represent the topology of the real tissue, in particular if we are working with a 3D bioprinter. The bioprinter works as a pen, and we are able to design the microstructure of our 3D structure in order to reproduce the topology of the real tissue. We have also to evaluate if we need a natural scaffold or a synthetic material. In the first case, we will not have any problems in cells and scaffold interaction, but we could suffer this problem in the case of a synthetic derived material. But in this case, we could have surface treatment in order to increase the cell adhesion or to release drug in situ. Moreover, the scaffold could be optically transparent. This is a very important aspect that has to be considered at the beginning of the experiment, because it's the key decision point to describe how to monitor, how to visualize our cells during the experiment. So if the scaffold is optically transparent, we can perform a classical monitor using an inverted microscope in bright field. Otherwise, we have to choose a different solution. A different solution could be use a fluorescent dye in order to to mark the cells at the beginning. But this choice has to be made before to start the biological experiment. Moreover, we have to consider the handling on the scaffold. So how to seed the cells onto the scaffold. Sometimes it's not sufficient to seed the cells on the top surface of the porous scaffold. Sometimes we need to force the cells to penetrate the pores of the scaffold, so we have to use a syringe, for instance. Or in other cases, it's just sufficient to have the cells in a liquid and the scaffold, maintaining overnight onto a shaker. Is it a ready-to-use solution? So once we buy the scaffold, do we have a ready-to-use product, or do we have to mix different solutions in order to obtain the scaffold? And in this case, are we able to varying the percentage of these solutions in order to obtain different stiffness? How much is precise the protocol considering the last aspect? Of course, in a ready-to-use product, we are not able to change the stiffness of the scaffold, but we are ready to start our experiment. We have to consider how to store our scaffold, because in sometimes it's okay to store at room temperature. In other cases, we need a -20 degree. Finally, we have to consider the post-processing, because it could happen that I would be happy to recollect all the biological material at the end of the experiment in order to do a post-processing, typically a DNA extraction. Sometimes the scaffold doesn't give you this opportunity. So we have to consider at the beginning in order to realize the procedure to do our post-processing. I would also add to these points also the time-dependent response that we have in a 3D structure. If we consider, for instance, to use a fluorescent dye in order to study the vitality of our cells, we need to consider that, for instance, in an hydrogel solution, we have to wait time in order to let the dye penetrate, diffuse in the inner region of the hydrogel solution, reach the cells and react with the cells. So the time that we will have to wait will be relevant if compared to the 2D solution. So all these procedures have to be studied before to start a biological experiment. So once we have made our decision, we need to choose the suitable environment to mimic the dynamicity of the real tissue. In this case, we have several platforms on the market. In general, the platform are characterized by the volume of the chamber. So a few hundred microliters is the volume of a microfluidic chamber. A millifluidic chamber has a volume of few mLs, and then we have the microfluidic chambers. In the last case, the microfluidic chamber are generally used to increase the number of cells or increase the biological material, or to test the mechanical properties of real tissue or mature tissue, such as endothelial vessel. And so they're not used in the setup of an in vitro models. So we have to choose between the microfluidic chamber and the millifluidic chamber. Here I summarize some of the main companies who are working in these fields. Of course, it's not an exhaustive list. It's only a summary. And here we have some of the main companies in the millifluidic chamber. The difference between the microfluidic and the millifluidic chambers are reported in this table. So a microfluidic chamber is characterized by a low volume. The low volume is in particular due to the reduced thickness of the chamber. So In this small volume, we have a saving of reagents, and it's easy to quantify the cell products over the time. Of course, the reduced dimension of the chamber forced to work at very low flow rate in order to avoid any damages onto the cells. If we consider, as I told you, that the fluid will cause a mechanical action onto the volume where it flows, we have to consider that this mechanical action is not going to damage the cells that we seed onto the wall. So we need to work at very low flow rate in order to reduce the shear stress. At the same time, we have some drawback. In particular, this low flow rate cause a low nutrients turnover. If a hair bubble is present in the chamber, it could be problems because the typical dimension of a hair bubble is comparable with the thickness of this chamber. So it could cause a stop in the flow or an increase of local pressure that could damage the cells. On the other hand, the millifluidic chamber is exactly the opposite of a microfluidic chamber. So we have an increase in volume. The volume is dependent because of the thickness of the chamber. So in this case, we are able to work a higher flow rate, maintaining a low shear stress, and this higher flow rate causes a higher nutrients turnover. At the same time, we are not able to easily quantify the cell products because we use a higher volume, and also we have to use more reagents. The hair bubbles are not so a big problem for a millifluidic chamber because typically the dimension of a hair bubble is not comparable with the dimension of the chamber. So depending on the goal of our experiment, we have to choose between a microfluidic device or a millifluidic device. And here I represent an example. Mimetis is a very well-known company who are selling these plates comparable in dimension with a standard plate, but where we have different microfluidic chambers. In this study, the researchers analyze different seeding densities or ECM composition, and also anti-cancer drugs in parallel. So it's a good solution to perform a high-throughput screening analysis. On the other hand, we could be interested in increase the amount of biological material in order to be correlated with the reality, and we could also interested in use the biopsy. The biopsy is the most complex 3D structure that we can use in vitro. So in this case, I present a study where our technology is used. I will describe the technology in the next slides. But here the researchers try to maintain a biopsy in vitro for several times and evaluate the effects of nanoparticles on this biopsy. So in this case, they refine their in vitro models using a millifluidic approach. So the choice between a microfluidic plate or a millifluidic chamber can be on-off. Otherwise, it can be a strategy where we have a high throughput screening at the beginning, performed using a microfluidic device, and then we can refine the model using a millifluidic chamber. Once we decided our technology, we have to evaluate how to mimic the organs crosstalk. Considering that the technology allows us to perform a multi-organ approach, so put several tissue in communications and study how they interact, we have to evaluate how to mimic this multi-organ environment. And here I represent two tissues, lung and liver. It can be an example of a drug that has to be inhaled, and we would be interested in studying the metabolism of the products of this drug once it has been metabolized by the liver. As you can see, the real parameters of these tissues are completely different. In case of a lung, it is a physiological barrier, so we have to consider, for instance, the exchange surface. In case of the lung, it's 200 meters square. In case of the liver, we could be more interested in the number of cells that we have in order to consider its metabolism. So how to scale these parameters in order to have a well-correlated in vitro models with the reality. So we propose a mathematical approach where we are going to choose the features that we want to mimic with our in vitro model. We have to look for the parameters. For instance, if we want to mimic the lung, we would choose the surface, the exchange surface, and we have to look for the real parameter, 200 meters square. Then we have an allometric approach. So we have to correlate this feature with the body mass. The allometric scaling will give us an information about the parameter for our in vitro tissue. In some case, the feedback of this allometric approach is feasible if compared with the technology that we have. So in that case, the design of our in vitro models is done, so we can start to build our biological model. In other cases, the allometric approach, the results is not feasible. So in that case, we have to choose for a different feature, or we have to choose a different technology. As I told you, an advanced in vitro model has several characteristics. We would like also to add some technical feature. In particular, it has to be monitorable on time. In particular, a black box, which is as close as possible to the reality, is not so efficient if you consider that it will give you feedback only at the end of your experiment. A very long experiment, you could have a endpoint analysis instead of a real-time monitoring. Moreover, in order to reduce the needs to rescale the parameters, the protocols that have been developed using a standard multiple plate system, we would like to use standard cell culture protocols and monitoring procedures. So in order to meet all these specifications, we have Ivytech. Ivytech mission is to increase the complexity of a 3D static model, adding a fourth dimension represented by the dynamicity of the model. So in our solutions, we have at least a flow of medium, which acts as blood in human circulation. This is not sufficient in order to have the refinement of the in vitro models, because we need to study the organs crosstalk. So our technology allows to perform 3D dynamic and multi-organ models, where we have the possibility to study the cross modulations of different tissues in communication because of the exchange of liquid. So this is the fifth dimension of the scenario. So we offer technology through a distributors network, and also services, technical support, realizing mathematical models such as the allometric approach that I described you previously, or studying the fluid dynamic environment in order to evaluate the fluid parameters a priori. Moreover, we have a collaborators network in order to build the biological model and offer as a service. This is our network of distributors in Europe and outside the European borders. Our goal is to become a reference partner to develop these advanced in vitro models. So in considering our solutions, we have small cell culture chambers based on the dimension of a single slot of standard multiple plate systems, where we are able to mimic the environmental stimulation of a real environment. we have the possibility to develop a 3D in vitro model in dynamic conditions. So we have the medium which acts as blood in human circulation. The dimensions of our chambers allow to use the same cell culture protocols developed for the standard plate. Moreover, the chambers are transparent in Z-axis, so it is possible to perform a real-time monitoring using a standard inverted microscope or even a confocal microscope. Finally, the top surface and the bottom surface of our chambers are made by a removable glass disc. So at the end of the experiment, it is possible to recollect all the attached biological material and perform a post-processing in a plate. Here we have our products. So we have the Lightbox 1 family, which is represented by a single flow bioreactor developed for attached cell culture models. In Lightbox 2, we have a removable and porous membrane, which replicates the standard transfer that we can find on the market in the porosity and in the material. This membrane divides in two sides the chamber, an apical chamber and a basal chamber, and we can work in liquid-liquid interface or air-liquid interface in order to mimic physiological or pathological barriers. Finally, we offer also a peristaltic pump, live flow, which is a very compact and low-weight system compatible with incubator environment and equipped with two independent motors with two removable heads. Each head drives up to 2 fluidic circuits, and so we are able to work with up to 4 fluidic circuits in parallel. Going deeply in our analysis, we have in the Lightbox 1 family, we have chambers compatible in dimension with the surface of a single slot of a 6-well plate, of a 12-well plate, or a 24-well plate. The reference system for the Lightbox 2 is represented by the transfer. So we have solutions compatible with the surface dimension of a transfer for a 6-well plate, 12-well plate, and 24-well plate. In this case, we suggest the use of a commercial standalone insert produced, for instance, by Nuke or Millicells, in order to house directly this kind of commercial solution in our chambers. Our chambers has a standard configuration, and then it can be also customized basing on the tissue specification. So, for instance, in this slide, I propose you our Lightbox 2 standard configuration on the top side of the slide, and then we can substitute, for instance, the top glass of our chamber with a hair-liquid interaction module. In the bottom part of the slides, you will see this interaction module, and opening this module, we are able to have a direct access to the tissue developed onto the membrane, and in case of a hair-liquid interface, we are able to inject a cosmetic compound directly onto the tissue and studying, for instance, its permeability through the tissue thickness. Here we have an overview of the hair-liquid interaction module on the left, and on the right, I focus my attention onto the holder. The holder is the system that we use in order to house the membrane. The standard configuration holder can work with membrane up to 100 microns in thickness, but we have also solution for very thick layer, such as a skin biopsy of 1 millimeter in thickness. And we have also solution for very thin products, such as the electrospun membranes, up to 50 microns in thickness. So we are able to customize the chamber basing on the applications that we want to develop. Focusing the attention onto the general aspect of our chambers, we have modularity. So each module, each chamber can be joined together with other modules in order to have communications between these systems. Each chamber can be focused to mimic a different tissue, so we will have a crosstalk of different tissues because of an exchange of liquid between these modules. In each module, we are able to see the 3D structure, a scaffold, already colonized by cells or which has to be colonized in situ. The scaffold can be a commercial version because of the compatibility of our chamber with the standard slot of the multiple plate systems, or it can be a homemade solution. If we work with a 3D bioprinter, we are able to directly print the 3D structure inside of our chamber in order to replicate the topology of the tissue. Once the tissue is mature, we can join together the chambers in order to have not only one tissue in dynamic condition, but a pathway in dynamic condition. The pathway can be joined with a fluidic circuit. The fluidic circuit is made by at least one reservoir, where we have the fresh medium or the waste, and tubes in order to connect the different modules of our systems. We have also a peristaltic pump. During the experiment, we are able to inject a drug in order to test its principal effect onto the target organ and its side effect onto the related tissues in just one experiment. As I told you, it's really important to have the possibility to monitor our cell culture, and it's possible to perform a real-time monitoring, sampling the liquid. So we use three-way valves, and because of the modularity of our systems, we can place the valves in different sections of the fluidic circuit in order to design a dose response curve. The sampling of this liquid can be performed avoiding the need to disassemble the chamber. Moreover, the chamber can be monitored using an inverted microscope or even an advanced optical imaging platform such as the Julie stage by IVTech. The dimension of the whole system are very compact and compatible with a small incubator. All the systems that I proposed you have to be placed in the incubator environment in order to control the CO2 concentration and the temperature level. Moreover, the head of the pumps, as well as the fluidic circuits, as well as the chambers, are autoclavable and reusable up to 10 times. In the last part of the presentation, I would like to focus the attention onto some applications of our products. So here we have the configurations that we can use. With the Lightbox one, we have just one configuration because we have one inlet and one outlet, so the liquid will flow from the inlet to the outlet. In Lightbox 2, we have several configurations. So we have a membrane, a porous and removable membranes, which divide the chamber in an apical compartment, the green part, and the basal compartment, the blue part. So we can have a flow of liquid on the top side of the membrane and a flow of liquid on the bottom side of the membrane. So we can work in a liquid-liquid interface, or we can work in a air-liquid interface, joining together the inlet and the outlet of the upper circuit. In some cases, our clients decide to use a perfusion configuration. So they use only the upper inlet and the bottom outlet, letting the medium flow from the top side to the bottom side of the chamber, passing through the pores of the membrane. So this configuration is suitable for all the solutions where we require to perfuse a very thick and porous scaffold, and it's the suitable application for a bone model. In case of, if we reverse the direction of this flow, we can maintain in floating condition cell aggregates or spheroids in the bottom chamber. In this case, we are going to use only the bottom inlet and the upper outlet, and the membrane represents a filter which avoids the flow of the biological material outside of the chamber. Here we have the universal color of Ivytech, if we want to cite a very famous slogan of a fashion company. We have the different applications of our chamber. So we have clients who are working in metabolism, who are performing permeability studies, who are testing new materials. In case of a new biomedical material, it could be important to test the biological effect of this material onto the cells, and in particular, using our chamber, it is possible to test the contact effect and also the release material effect. It is possible to develop a 3D model starting from stem cells or cell lines or primary cells, or also using a very mature tissue such as a biopsy. Finally, the multi-organ approach can be used to develop a disease model. If we want to go deeply in our analysis, here we have the tissues used to develop these applications, and we have papers or publications about these different studies. I would like to propose you some literature, some references, and the color are related with the different applications. At the end of the webinar, we will be able to share with you the presentation. And I would like to focus my attention on this study where a client decided to use a biopsy in our chamber. So when these clients start to talk with us, they already have characterized their nanoparticles for drug release in situ. And in particular, these nanoparticles have to act onto muscle cells. So they had two different publications with murine muscle cells and with human muscle cells. In both these studies, they obtain a very good results where the nanoparticles penetrate the structures and release their drug as in their idea. Before to start the animal test, they decided to refine the in vitro models using an advanced in vitro models. And in particular, they decided to use a biopsy, a muscle biopsy, in order to have a very complex 3D structure in order to test the effect of their nanoparticles. So they decided to use solid muscle biopsy from rat. And the first problems that they had was related with the phenotype of the biopsy cultivated in vitro. So as you can see, on the left, we have the control. On the right, we have the phenotype of the biopsy after 24 hours in static conditions. As you can see, we have a completely loss of the phenotype after this short period. So the idea was to place the muscle biopsy in a Lightbox 1 and use the fluidic circuit in order to provide nutrient and fresh oxygen over the time. And they obtained a very good results because after 24 hours, the phenotype of the biopsy cultivated in a dynamic condition was similar to the control. Moreover, they also increased their analysis, evaluating after 48 hours a good preservation of the microstructures inside of the muscle biopsy if compared with the conventional culture. So at this stage, the client was ready to test its nanoparticles. So they inject the nanoparticles in the fluidic circuits and evaluate the effects. But they obtain a surprising feedback. In the advanced in vitro model, the nanoparticles were not able to penetrate the biopsy. In particular, these nanoparticles, after a few hours of incubation, were concentrated on the peripheral region of the biopsy, and so were not able to penetrate the inner region of the muscle, releasing their drug. So they decided to stop the experiment and restudy the nanoparticles from a chemical point of view. The question is it a scientific failure? Well, Of course, they didn't obtain the results they hope, but they also evaluate the expenses that they have to perform in order to obtain these results, and they compared these expenses with a typical in vivo analysis. As you can see, if they performed an in vivo analysis, they would spend even 6.5 times more than the effective expenses. So they reduce the cost of their study. They also claim that no animal was killed for this research because they use animals, they use cavia that were previously used in different studies, so they had to be sacrificed at the end of these protocols. In order to give you the conclusion, it is possible to reduce the number of animal models using an advanced in vitro models. We are not talking about substitution, we are talking about reduction and refinement. The refinement of an in vitro models need an upgrade of the technology and the procedures. We have 3 main steps. We have to choose the right scaffold, so the right 3D structure. We have to choose the suitable fluidic environment. And in case of a drug test, we have to consider how to mimic the cross-modulation of the different tissues. The Ivytech products meet all these specifications and are user-friendly products that allows to study the cross-modulation of the tissues as a response to an exogenous stimulus. So it's all for the presentation. Thank you for your attention, and if you have a question, I will be happy to answer.
Tommaso, thanks a million for this great presentation.
Thank you.
I think a few questions came in, and the first question, I think it might be only a question of this time at the moment. Which system would you recommend for any lung in vitro models which might be usable for coronavirus research?
Well, you know, the coronavirus research needs has two different applications, two different specifications. Sorry. The first one is that we need a mature tissue in order to test the effect of the coronavirus on it. And then we need to be able to perform a virus study. So we need let's say, the safety degree of the environment. So I know that there are some reconstructed tissue about the lung epithelium. So I would start my test using this ready-to-use material, also because the reconstructed tissues sometimes are also ready from a legal point of view in order to be an alternative to the animal models. In case of the coronavirus, we can use this reconstructed tissue in order to study the biological environment. Then we need let's say a fluidic platform in order to mimic let's say the exchange of material across the lung epithelium. In our case, for instance, the Lightbox 2 equipped with the hair lipid interface module could represent a solution. Of course, then we have to let's say to inject the virus. We have to let the virus penetrate and react with the tissue, and maybe having a modular approach, we could also test the effect onto other tissues related with the lung epithelium. So this could be a first platform in order to test, for instance, the drug effect.
Okay, I think thanks. Just another question came in, and I have to check whether I can make it a little bit. Yes, I think that's better. Have you ever done a test with muscle? In general, I think you show this example at the end of your presentation with the muscle tissue.
Exactly. Yeah.
And have you ever done a test with muscle cells and differentiation under flow conditions?
The muscle tests have been performed using the biopsy, so they have already a differentiated tissue. They don't. Our clients didn't use different cells in order to test the muscle solution in our chamber. So the example for the muscle for us it's represented by the biopsy.
Okay, I think that's a few questions going in this direction. The next one is, can I study different pre-treated cells in the same experiment in order to see the different behavior?
Yes, of course. It's possible to have different pre-treated cells. It's possible to have in different experiment, or we could have also in different chambers in case of we need to evaluate the crosstalk of these different pre-tested cells. Yeah. Also, if we consider a multi-organ applications in case of pre-treated cells, we have also to consider the medium which we are going to use in our applications. In particular, if we use, for instance, 2 or more tissues, we have to use a common medium. Where if we use a Lightbox 2, the Lightbox 2 has 2 different compartments, and in these slides you can see better. We have 2 different compartments, 2 different circuits. The communications between these circuits will be only through the membrane pores. So here we can have in the green part, we can have the medium number 1, and in the blue part, we can have a medium number 2. So all the chambers connected in series with the apical chamber can be filled with the liquid number 1 or with the liquid number 2 in case of connection with the bottom chamber.
Okay, I think thanks a lot. By the way, I just want to add something. I think my colleague and partner, Mr. Steep, I think he is well known and our developer for media composition, so he might be very helpful in the discussion also together with Tommaso to find the best media composition for these kind of multi-organ in vitro cell systems, because the media requirements might not be the easiest one. Then there's another question which came up. What is your main application, or what was your main application last year? Which type of systems and models you are selling?
Yeah, the Ivytech is an engineer company, so we produce the chambers and then our clients develop the applications. The most advanced applications using our chambers is represented by circulating biological material, such as it can be lymphocytes, which circulate in the fluidic circuit and interact with different tissues in attached condition. So we have example of lymph nodes model, of bone marrow. So these are the most advanced applications. So having a circulating material which has to act with a 3D structure in attached condition.
Okay, I think that the next question goes in a very similar direction. Do you have already new ideas in the pipeline for this or next year? Some new improvements, whatever?
Yes, if we don't consider the coronavirus, we had to release at least 2 products on the market at the end of this year. One is related with a new version of the pump, and one is related with an increase of parallel degree that we can obtain using our chamber. So we are studying both on the fluidic aspect as well as the pump.
Okay, another question is go more in detail again. Can your systems be used for 3D angiogenesis models?
Could you repeat 3D?
3D angiogenesis models. That means normally a co-culture of epithelial cells and fibroblasts.
Yeah, we already have papers where it has been studied the development of a mature tissue starting from stem cells, in particular in bone, and they use fibroblasts in order to develop this mature model. So I think I already reported the literature link here, so it's ready to be read.
Okay. Okay, I think, by the way, also we will answer the questions again also by email, as well as this webinar was recorded, so you will receive also a record of the webinar, so you can listen again and check the slides Tommaso showed. There's another question: how far is your microfluidic platform from entering high throughput screens, and what are the main obstacles?
Well, I think that we have to consider the slides where we presented the microfluidic device and the millifluidic device. Here we have different structures, different ideas. The high throughput screening can be performed with a very small compact products where we have several chambers in parallel. I think that this millifluidic device will not be a suitable solution for high throughput screening analysis. It's always to be used as a refinement of the in vitro model. So I would say that the high throughput screening will be performed in the next future using a plate system or a microfluidic chamber, and then once we want to increase the correlation with the human reality, we will add biological material, we will use a more complex 3D structure, and then we will use a millifluidic device such as the products we offer.
Okay. Great. Another question goes again in direction of angiogenesis. Is it possible to apply a gradient to the tissue within the Lightbox too?
We have a client who is working with angiogenesis in Lightbox 2, and in particular, she is working with this kind of configuration. So a sigmoidal throw bottom side to the top side, and here she has the 3D structure starting from let's say stem cells, and then they studied the angiogenesis of the 3D structure. And here, of course, we will have a gradient, which is not a controlled gradient, but can be studying basing on the flow, the inlet flow, and the fluid dynamic of inside of the chamber.
Okay. I think there are a few other questions which we will answer, I think, later. I think, do you have any recommendations which hydrogels or scaffolds can be used together with your Lightbox systems?
In my opinion, the considerations that have to be performed considering the hydrogels have to take in consideration the post-processing. I mean, in some cases, the hydrogels does not permit the recollection of the biological material, and this is a drawback. If you consider a very standard hydrogel, which could be, for instance, the matrigel, at the end of the experiment, you are able to dilute the matrigel solution in order to obtain to recollect the biological material. So as far as I know from my clients, the key decision point in an hydrogel solutions is represented by the possibility to recollect the cells at the end of the experiment.
Okay, I think one comment from my side, there are a few synthetic hydrogels in the market now where you can, I think, define the stiffness, which are, I think, smart ones, so that you just put the Lightbox into a fridge, and at 4 degrees the hydrogel is a liquid, and if it's at 37 degrees it's a solid material. So these systems can be used. I think if you like to get more information about that, just let me know. I think that's the main questions which came in. Just want to say again, thanks a lot for everyone. I think there seems to be no question at the moment. If you like to get, I think, more information, please send us an email. I think we will be happy to answer this also later. I also like to say, Tommaso, thanks again.
Thank you for your kind invitation, Peter.
That's a pleasure, and I like to just remember you that we have, I think, a series of webinars which is all around that we ask you the question, can an in vitro cell culture be as good as in vivo? And we will have the next session, the next webinar on April 23rd, which is about tissue isolation and cell viability. I think it's presented by my colleague and partner, Dr. Lothar Steb. So you're welcome to join us again. I thank again everyone for attending and wish you a safe and healthy future at the moment here in Europe or wherever you are. Thanks a million and take care. Bye-bye.
Thank you for your